Assalamualaikum and good day everyone. So today we will continue with the next topic, which is on transcription factors and transcriptional control. So we know that binding of DNA transcription translation is controlled by basically lots of factors, okay, and they start off with the transcription factors. So it starts off with DNA binding, and we know that transcription factors, they are basically protein that regulates transcription of genes, whether to turn it on or off or up or down. So lots of different transcription factors are needed to ensure right proteins are made at the right place, okay, at the right time. So let's have a look first, okay, at how DNA binding, okay, occurs, okay, by general transcription factors. So basically, general transcription factors, the function is to aid in binding of RNA polymerase II to the promoters. And how was this done? So some of them, as we know from last week, again, recognize specific sequences in the DNA, and once bound, they basically act as a platform, which of course, okay, in turn helps the RNA polymerase to attach to the promoter site, kind of like preparing RNA polymerase, okay, to bind to it. So the assembled group, okay, of the general transcription factors, okay, plus the RNA polymerase is sometimes called the transcription initiation complex, and usually they are referred to as such. So general transcription factors are general in that basically they are required to begin transcription of every gene that is transcribed by the RNA polymerase, because without which then RNA polymerase couldn't attach and you couldn't have initiation of transcription. So there we have it again. So for example, stretch of gene, again, from prime to 3 prime, you have your promoter sequence over here, you have your transcription start site somewhere here. So whatever happens is that, again, general transcription factors have to bind to the promoter first, recognize it again, then only basically recruit some more of the transcription factor. We know that after about 3, okay, of them have bind, again, then only the other 3, okay, will get RNA polymerase again, and then they form the transcription initiation complex. Now, how about by the specific transcription factors? So there are also specific transcription factors involved in DNA binding, and they are regulated, okay, they are the ones, okay, that mostly regulate, okay, most eukaryotic genes. What are these specific transcription factors? They are usually the one that control the transcription of specific target genes, but not all of the genes in the genome, okay, only certain specific target genes. Why are these specific transcription factors needed? Because for many genes, especially this is mostly in animals, general transcription factors alone are not enough and will not cause much transcription. This is also true in most cases of plant. So instead, the right set of specific transcription factors must also be present to put the gene in its on state, to turn it on, to have it expressed. Now, this means that the gene is expressed only under certain condition, whereby, okay, it is only signaled by the specific transcription factors. So kind of like your typical specific transcription factor is a DNA binding protein that recognizes a target DNA sequence. So whenever it's find the sequence, so transcription factor will bind to the DNA, and the bound transcription factor affects the binding of RNA polymerase at a nearby promoter. And of course, sometimes not so nearby depending on the location. So first and foremost, before we move further, this will be part 1, okay, of transcription factors and also transcriptional control. So in second part, we will have a look further, but let's have a look at the transcription factors family first. So basically, these are the common types, okay, of motifs that are responsible for binding to DNA, which can be found in different transcription factors. So there are crucial features, okay, which must be atomic contact between protein residues and bases and sugar phosphate backbone of the DNA. That's why, okay, it's a protein, so it could bind. And most contacts, okay, are in the major groove of DNA, meaning that, okay, 80% of regular proteins can be assigned, okay, to one of the 3 classes of the transcription factor family, such as helix turn helix, HTH, zinc finger, or leucine zipper, BZIP. So in addition to the DNA binding domains, these proteins usually possess other domains that interact with other proteins. Of course, okay, it's basically the ones, okay, that is involved, okay, in the initiation or recruitment of RNA polymerase, but it also basically interact with other proteins apart from the transcription. So let's have a look first at the helix turn helix proteins. So it's actually quite easy to remember, right? And there's only 3. So it has 2 repressor proteins, CRO and CI, and the E. coli catabolite activator protein, CAP. Remember the CAP again in your prong? So all these bind as dimers to diet symmetric sites on their DNA, and all of them contain 2 alpha helices separated by a loop with a beta turn, and the C terminal helix basically fits in major groove of the DNA, whereby the N-terminal helix stabilizes by hydrophobic interaction with the C-terminal helix. So the N-terminal is actually towards the 5', okay, C-terminal also towards the 3', okay, more or less, if you could imagine that, okay, but basically their location is everywhere, okay, but you have the ones, okay, that starts it, okay, and then the ones that ends it. So recognition of DNA sequence involves the size of the base pair that face the major groove. So this is more or less, okay, the helix turn helix protein structure, okay, and basically how it interact with your DNA later on. So this is the diet axis of the symmetry. Now, second one that we will have a look is the zinc finger protein. So it is now known that the zinc finger protein exists in nearly all organisms, including prokaryote. And there are two main classes of zinc finger protein, okay, C2H2 and CX. X meaning that, okay, basically you will see in a while, okay, as you can see over here. So C2H2, the most common one, okay, consists of cis X2 cis and his X3 his domains separated by at least 7 to 8 amino acids. So a finger protein typically has a series of zinc fingers, so that's why it's called a zinc finger protein. So the consensus sequences of a single finger is this, okay, cis X2, 4 cis X3, phenyl alanine X3, leucine X2, and his X3 his. So this is histidine, this is cysteine, okay, for those of you who don't know what it means. So the CX domain, okay, the ones here, okay, consists of 4, 5, or 6 cis residues separated by various numbers of other residues. So this is more or less the structure of zinc finger protein. So this is the typical, okay, C2H2 that you would see, and this, okay, would probably, most of the time, okay, you could also have the C2H2, okay, in this form, okay, but sometimes, okay, this could also represent the CX as well. So for the C2H2 type zinc finger, usually it forms a folded beta strand and an alpha helix that fits into the DNA major groove, okay, the B that you see in the previous slide. So the CX type zinc finger consists of two mini domains of 4 cis ligands to zinc followed by an alpha helix, okay. The first helix is of course the DNA. So the recognition helix, okay, is basically the ones, or the second helix, that packs against the first. And more or less, okay, this is how it looks like. So don't worry too much on the structure. Usually I don't really ask about the structure to draw or whatever, because you need to be very good in protein, even though I know you are learning about protein engineering, but this is quite complex, okay, to understand. But basically this is just structure and how it looks like. Alright, and the last one, okay, is the leucine zipper protein. So it is also found in nearly all organisms, including prokaryote, and the characteristic feature is a 28 residue sequence with leucine every 7th position and basic region. Basic, not your basic, okay, but base region. So this suggests amphipathic alpha helix and a coil dimer. So leucine zipper proteins, okay, or called B-zip, dimerize either as homo or hetero dimers. So the base region is of course the DNA recognition site, and the basic region is often modeled as a pair of helices that can wrap around the major groove for transcription. So when it is in homodimers, okay, it recognizes diet symmetric protein. If it is in heterodimers, it recognizes non-symmetric DNA. So apart from that, okay, B-zips are also referred to sometimes in their own names, okay, kind of like if you see, okay, in certain papers, when people refer to as FOS or June, okay, they are classic B-zips. So more or less, this is the structure of leucine zipper protein, kind of like nice, right, diets, okay, in terms of its structure. Alright, so for the leucine zipper proteins, so dimerization usually allows the juxtaposition of the DNA binding region, two of each of the subunit, and leucine zipper will form an amphipathic helix in which the leucine of the zipper on one protein could of course protrude from the alpha helix, okay. So if you were to see this, okay, protrusion, okay, is as such, okay, and of course, okay, interdigitate with the leucines of the zipper of another protein in parallel to form a coil domain. So the region adjacent to the leucine repeats is of course highly basic in each of the zipper protein and could comprise a DNA binding site, okay, and this actually helps or increases, okay, the binding for transcription. So the two leucine zippers in effect form a Y-shaped structure in which the zippers comprise the stem and the two basic regions bifurcate symmetrically, okay, to form the arms that bind the DNA. And of course, this is known as the bizip structural motif, as you can see over here, kind of like your antibody, but this is a zipper protein, so it forms such as this when dimerization occurs. Now, we go back again to the helix loop helix protein. More or less, okay, for the helix loop helix protein, so it also forms amphipathic motif, which of course identified in some developmental regulators and in genes coding for the eukaryotic DNA binding protein. So proteins that have this motif have both the ability to bind DNA and also to dimerize. And bear in mind, not all proteins have this. So they could share a common type of sequence motif. Okay, for example, a stretch of 40 to 50 amino acids containing two amphipathic alpha helices separated by a linker region, which is a loop of varying length. So proteins in this group, okay, helix loop helix proteins, form both homodimers and heterodimers by means of interaction between the hydrophobic residues on the corresponding faces of the two helices. So helix loop helix proteins, there are two general groups, class A and class B. So class A consists of proteins that are ubiquitously expressed, so including mammalian E12, E47. So class B consists of proteins that are expressed in the tissue-specific manner, so including mammalian MyD, okay, MyF5, and mygenin and MRI4. So these are a group of transcription factors involved in myogenesis, okay, or the MRF. So a common modus operandi, okay, which means how they operate, okay, for a tissue-specific helix protein, may be to form a heterodimer with a ubiquitous partner. As such, okay, so it forms the DNA binding okay, to form heterodimers. Alright, so I guess that would be it, and then you guys move on to the second video, whereby we will move okay, to some other transcription factors.